Hello and welcome back to Chapter 3 of Human Physiology. So last time we finished up Chapter 2, which was focused mainly on molecules. So we talked about inorganic molecules like water, and then we talked about organic molecules like proteins and lipids and carbohydrates and nucleic acids. Basically, the types of molecules that are going to comprise a cell. Well, so appropriately, Chapter 3 is all going to be about the cell itself. We are going to see how a lot of those organic and inorganic molecules that we talked about in Chapter 2 can actually contribute very much to the functioning of the cell and eventually to the physiology of a person. So you can see the checklist here. This particular video, as with the others, is going to just focus on the first item on this checklist, and then subsequent videos will tackle all the rest. So let's go ahead and get into it here. So this is a picture that comes directly from your book, and this is a really nice microscopic image of a cell that is going through the cell cycle. Now, we're not really going to focus too terribly much on the cell cycle here for reasons that will become clear as we kind of make it towards the end of the chapter, but this is a really nice picture that kind of demonstrates how all of those organic molecules that we talked about in Chapter 2 are coming together to kind of put forth a very important cell function. So everything that you see in blue here, these are all of the chromosomes that have been duplicated, condensed, and are now being separated. So those are made up of nucleic acid and protein. In green here, we have microtubules, which are protein filaments that attach to the kinetochores of the chromosomes and start to pull them apart. In red here, we have the actin cytoskeleton that is just underneath the plasma membrane. It's also a protein. The plasma membrane, of course, is made up of lipids and proteins and carbohydrates. So this is a really nice picture in as much as we're not really going to be talking about the process of cell division itself, but it really demonstrates nicely how all these things work together. So you will recall from Chapter 1, when we discussed levels of organization, cells are just going to be the accumulation of many, many types of inorganic and organic molecules. So they are all working together in some meaningful way. We also mentioned that in kind of your standard human person, there are about 30 trillion total cells and about 200 types of cells. So these 30 trillion cells and 200 types of cells are the result of cell division being mitosis and then also differentiation. That's how we get different types of cells. We also covered how when you were conceived at fertilization, you existed as a single-celled organism called a zygote. Many rounds of mitosis produced more and more and more cells, so that's how you go from 1 to 2 cells to 4 cells to 8 cells and so on. And then differentiation causes cells to become specialized into things like brain cells, muscle cells, and so on. So something that we're really going to start focusing on here is that even though we have 30 trillion cells in our body, and even though there are 200 different types of cells, the cells are all going to have quite a few different things in common in terms of having a plasma membrane, having nucleic acid stored in the nucleus. But a lot of these cells, kind of for our money, when we look at them, are going to look very different from each other. So this picture that you're looking at right here, on the left, you see a sheet of squamous epithelial cells, which you might find in the skin or in the respiratory tract or in the digestive tract. And then on the right here, you have a neuron, which is a cell that forms nerve fibers. So clearly these two cells look just about as different as they could from each other. I say these two cells. This is one cell on the right and then many cells over here on the left. But these types of cells look very different from each other, and what we're going to learn here is that a big reason they look different is because, in terms of structural difference, is because they ultimately have different functions. They look different because they have different things they are trying to accomplish, all for the sake of homeostasis and our normal physiology. And then, as you can kind of imagine, based on what we've said, the reason these cells ultimately look and function differently, it's all the result of that differentiation process, which is a process that is laid out and acted upon as dictated by what is in your DNA, what is in the genetic instructions. So even though this is a physiology class, and you probably didn't, maybe you did, but you probably didn't imagine that we'd be spending a whole lot of time talking about individual cells. You probably thought we'd be mostly talking about organs. Well, the thing to make sure you really understand moving forward is that cells are the basis of all human physiology. I mean, it's all fine and well to talk about the heart as an organ, the brain as an organ, the liver as an organ, but these things are made up of cells, and it is the individual function of all those cells acting together that is really doing the heavy lifting in human physiology. So that is something definitely to keep in mind. One example of this, if you consider the nerve cells, the neurons and the like that make up, say, your brain, your spinal cord, and all the different nerves in your body, they are going to, as individual cells, be acting as control centers if you're talking about the brain and the spinal cord, input and output signals if you're talking about the nerves. So these are individual cells, so they are actually capable of acting kind of in a major role when it comes to the homeostasis loops that we talked about in Chapter 1. Another perfect example, the cardiac muscle cells of the heart. Don't forget the heart is an organ and it's made up of cells. These individual cells, each one of those cells provide these strong contractions that help to pump blood throughout the body to make sure that we get all those nutrients and things that we talked about that we need back in Chapter 1, like oxygen and food molecules and micronutrients and things like that. These individual cells provide the contractions we need to deliver those things to all the different cells in the body that need those things. In this case, all those cardiac muscle cells acting together are effectors because the heart can respond to output signals that are sent from the brain. So with that being said, one of the major topics in this chapter, as we kind of focus in on the cell itself, one of the major topics we really want to focus on here is the separation between what is inside the cell versus what is outside the cell. So you will recall from Chapter 1 that we briefly mentioned that when you're talking about bodily fluids, there's basically a compartmentalization of an intracellular fluid being all the fluid inside the cell, and then an extracellular fluid, which is all the fluid that is outside the cell, meaning the interstitial fluid and the blood plasma. So the thing that actually separates these two body fluid compartments from each other are plasma membranes. So every cell in your body is going to have a plasma membrane. So if you look at this picture here, if you look at these kind of yellowish white looking cells in blue there, you can see their nuclei. So the borders of each one of those cells are made up of a plasma membrane. So that's what keeps the intracellular fluid in and the extracellular fluid out. Similarly, for these blood vessels here, the wall of the blood vessels are made up of cells called endothelial cells. So ultimately, that is another thing that keeps the extracellular fluid and intracellular fluid compartments separated from each other. So we talk about the plasma membrane like it's some kind of impenetrable barrier, like everything in the intracellular fluid is supposed to stay put, everything in the extracellular fluid is supposed to stay put. And that's not really the truth. So the cell membrane is a restrictive barrier, but it is not an impenetrable barrier. It's not some concrete wall that is never to be crossed. So the common terminology is that the plasma membrane is said to be selectively permeable, meaning the membrane will allow certain types of things to pass through while greatly restricting the movement of others. So it might be more appropriate to think of it kind of as a wall that has pores in it, right? So certain things can pass through, but other things cannot. So to that point, the cell membrane is actually a very dynamic and pliable structure. Like I said, it's not a concrete wall. So the ability of molecules and atoms and other sorts of things to pass from the extracellular fluid to the intracellular fluid and vice versa actually underlies a very fundamental need in our homeostasis. We actually absolutely have to be able to transport materials back and forth from ECF to ICF and ICF to ECF. So we really need the plasma membrane to not be an impenetrable barrier. We need it to be dynamic, as we are saying. So really, before we can understand how these sorts of things occur, we need to actually look at the structure of the plasma membrane, which is something that we kind of touched on towards the end of chapter 2 when we started talking about lipids, but we really did not see the payoff for that. So let's go ahead and get into that. You will recall that the plasma membrane, the cell membrane, is majorly made up of a type of lipid called a phospholipid. So a phospholipid is a type of diglyceride, meaning it contains a glycerol backbone with 2 attached fatty acid tails, and then where the third fatty acid would go if it were a triglyceride, we instead have a phosphatidylcholine group. So this makes the phospholipid very interesting because it is what we call amphipathic, meaning that it is not purely hydrophobic and it is not purely hydrophilic. It actually has partial characteristics of both. So it ends up with what we call a hydrophilic head, which is where the polar phosphatidylcholine group is, and then it has what we would call hydrophobic tails, which is where the fatty acid chains go. So if we're thinking about constructing a cell membrane out of phospholipids, the thing that we need to be concerned with is something that we very much made a big deal of back in chapters 1 and 2, the whole hydrophilic hydrophobic thing, in terms of for a hydrophilic or hydrophobic molecule, they are defined by their interactions with water. So we really need to consider how the phospholipid will potentially interact with the water in both the ICF and the ECF if we are going to try to construct a plasma membrane out of phospholipids. So let's take a look at this. So let's say that we have no idea how a plasma membrane is put together, so we're just going to kind of tinker with this. So what if we tried to make a plasma membrane out of a single layer of phospholipids, kind of like what you see here? So on one side of the phospholipids, you have the extracellular fluid, and on the other side, you have the intracellular fluid. So keep in mind, there is water in both the intracellular and extracellular fluid. It's the major component of both. So as you look at this single layer of phospholipids, can you identify what the problem is here? Well, clearly the problem there is that since there is water in both the extracellular and intracellular fluid, one side of the molecule will be happy in terms of those red polar head molecules. They will be happy because they are interacting with water. That's exactly what they want because they are hydrophilic. But the tails are not going to be very happy because they are hydrophobic. They are being forced to interact with water even though they do not really want to. So it is for this reason that we do not build plasma membranes out of a single layer of phospholipids. We actually make a double layer, what is called a bilayer of phospholipids. So the way we do this is we stack the 2 layers kind of flip-flopped on top of each other so that the tails are pointed towards each other and the heads are pointed out towards the aqueous environments of the extracellular and intracellular fluids. So everyone should be happy in this arrangement. The polar heads on both the top, on the extracellular side, and on the bottom, on the intracellular side, the heads should be satisfied because they are hydrophilic and they are interacting with the water in the 2 fluids. And the tails should be happy because that hydrophobic core, basically the middle of that sandwich, if you want to think of it that way, that hydrophobic core completely excludes water, meaning there is no water in the middle of the phospholipid bilayer. So the tails are happy because the only thing you find there are hydrophobic molecules, like the tails and like a couple other things that we will talk about later. So another thing that we should consider is that the plasma membrane, as we said, is very dynamic and very pliable. Another way of saying that is it is very fluid. It's not like a congealed solid mess. It is actually closer to being a liquid than it is a solid. And this characteristic is going to be critical for allowing molecules to pass back and forth. So as I said, if the membrane was kind of a congealed solid, kind of like butter or lard, which are other types of lipids, then it really would be an impenetrable barrier. If the membrane were completely solid, nothing's going to get through, right? So the membrane ends up being fluid for a couple of reasons. So number one, and we can kind of go backwards so you can see the picture again, one of those phospholipid fatty acid tails is of the unsaturated variety. So if we go backwards here, you can see that fatty acid tail on the right contains a double bond, which, as we indicated in chapter 2, that is going to cause a kink in the ordinarily linear fatty acid tail, so this causes the phospholipids to kind of spread out a little bit. It's kind of like swinging your arms out to the side. It's going to give you some extra elbow room. So because of that, the kink allows adjacent phospholipids to space out, meaning the density of the plasma membrane will be less, and it will be much more agreeable to being a fluid rather than a solid. The other reason is that hydrophobic core contains a lot of cholesterol. Cholesterol molecules, a type of steroid that we talked about in chapter 2, does the same sort of thing. It wedges itself in between fatty acid tails of adjacent phospholipids and spaces them out even more, keeping the membrane even more fluid. So yet another reason why we should not just automatically conclude that cholesterol is a bad thing. We need cholesterol for two reasons. We need it because it is the starting material for synthesizing steroid hormones, and we need it for this reason too. It's a critical component of the plasma membrane. So that is going to do it for this particular video. So join us next time and we will start talking about other things that you find in the plasma membrane. We'll talk about some of these carbohydrate chains that you find, but the main thing we're going to talk about is all the different membrane proteins. And as we are going to see, it is those membrane proteins that are going to allow us to freely transport molecules back and forth. So next time we will talk about membrane proteins and membrane transport. Hope you will join me next time. Thank you for your attention, and